Hello wonderful person, this is Anton, and today we're going to be discussing one of the biggest mysteries in science, the origin of life on planet Earth. And specifically some of the recent discoveries and some of the most recent research that tries to identify some of the earliest genes that existed on the planet and basically try to figure out how life might have started. Now, just so that we're clear, we still have no idea exactly how it happened, but because of these two recent studies we're going to be discussing today, researchers got just a little bit closer to understanding the process. But first, let's actually start with some of the things we already knew, and specifically let's start with LUCA. And that's because for decades researchers have been able to trace the history of life back to a single point biologists often refer to as LUCA, the last universal common ancestor. Basically some kind of a protocell or some kind of an early life that became so successful on the planet that it eventually evolved into bacteria and archaea that then obviously evolved into everything else. But there's always been a problem here, because in this case, LUCA, which very likely evolved approximately 4.2 billion years ago, or approximately 300 million years after Earth formed, was already an incredibly complex cellular organism. It already had a genome, it already built proteins, and it already had functioning membrane. And we know all of this based on the genetic analysis of a lot of bacterial and archaeal cells, which seem to share certain genes that must have come from LUCA. And so we know that this was a single-celled, prokaryote-like organism that used its own genetic material or its own DNA, and also very likely relied on the ATP for energy, and most likely lived in very enriched environments, such as near hydrothermal vents, in order to basically survive and in order to thrive. But since there are no actual fossils of LUCA, we don't really know much else. And that means that LUCA was not the first life, and instead was a very successful cell that eventually evolved everything else. And that means that we have no idea what the first life was like. And also because this was 4.2 billion years ago, the genes from this organism experienced what's known as horizontal gene transfer, or basically became shuffled in a lot of different organisms alive today. But even back in 2016, at least one study was able to identify 350 individual genes that most likely came from LUCA, essentially confirming that this was an anaerobic organism, not relying on oxygen, and was also able to fix its own CO2 for energy and to build its own proteins, and might have also been able to fix nitrogen to create ammonia. And moreover, it also contains certain proteins that seem to protect it from viruses, implying that viruses were already attacking cells and were already driving evolution. But this is still an extremely mysterious part of history, and there are still a lot of unanswered questions about the overall transition from the very simple cells to much more complex cells. Something that we didn't really know much about until some of these most recent studies. And that's because these studies now provide us with new tools to try to analyze LUCA and even analyze life before LUCA in order to understand what happened during those first few millions of years. And so basically in evolutionary biology, one of the biggest mysteries is of course this pre-LUCA era. The period before LUCA was evolved, and the period that we know absolutely nothing about. And well, up until recently, it was practically impossible to analyze any of this, because once again there are no fossils, and even trying to analyze genetics was extremely challenging. But in this recent study, researchers found at least one intriguing technique that allows us to use this genetic fossil that's technically hidden inside our own DNA and basically DNA of everything around us. And so in this new study from 2026 by researchers Aaron Goldman, Gregory Fournier, and Betul Kassar, researchers focus on something referred to as universal paralogs. And in biology, the term paralog refers to a bunch of related genes within a single genome, and normally they're produced as a result of duplication. And the best example here is actually hemoglobin, or specifically hemoglobin genes. Humans contain 8 of them, and all 8 of them are just copies or descendants of the original hemoglobin gene that appeared approximately 800 million years ago. And so here the formation of this protein containing iron that then allows the blood to transfer oxygen first appeared 800 million years ago, and then eventually evolved and copied itself with many different species containing different versions. And once again, humans contain 8 of them. These are essentially paralogs. But the point in this research was to actually try to identify some kind of a universal paralog. So basically not just hemoglobin that only applies to animals containing blood, but something that's even more universal that pretty much all life on the planet would contain. And so here they were looking for a family of genes that seem to exist in at least 2 copies in almost every living organism on the planet. And that's because by finding these universal paralogs, or these genes that were basically copied into every living organism, we would then be able to identify original genes that must have duplicated even before LUCA existed. And so these very rare genes effectively allow scientists to peer back in time and to try to see what life was like before LUCA was around, and very likely right after life became extremely common, or at least extremely successful, naturally because these genes never died out and remain with us ever since. And here researchers do discover something. They found that all known universal paralogs seem to involve just 2 separate tasks. First one is building proteins. Okay, that one is kind of obvious. You need proteins for life to exist. But the second one is a little bit more exciting. It involves moving molecules across cell membranes. And that of course suggests that both building proteins and moving stuff across cell membranes seem to be some of the first and some of the most successful biological functions to ever evolve. Or I guess technically you can even define this as life. Maybe that's actually what the definition of life should be. And that, by the way, connects to this other question we tried to discuss in a previous video you can find in the description. Because even today it's not clear what exactly life is. But to make this even more interesting, researchers behind the study even performed an intriguing experiment. And specifically, they actually resurrected one of these ancient proteins from one of these pre-LUCA families, mostly focusing on the protein you see right here. And well, they discovered that even this ancient simplified protein was still able to attach to a lot of cell membranes and even interact with protein-making machinery, suggesting that this was indeed a universal protein that very likely exists longer than anything else. And this by itself is quite incredible because it definitively proves that cellularity, or the idea that the life seems to be contained inside a membrane, seems to have been established on Earth extremely early on, and was possibly the main reason life was so successful. Obviously because membranes in this case provide a lot of protection. And so it was probably the membranes that seemed to play the biggest role in allowing the life to become so successful. But this is just the first study and the first experiment, so we don't really know much else. But we do know something else from a separate study on RNA molecules. And this is based on a study led by Philip Holliger that was recently published in Science. And so basically here, even though the paralogs tell us a little bit more about how cells worked, it doesn't entirely tell us why cells existed or how the first genetic material even started to copy itself. And that's of course the other major problem. How did the genetic material evolve and where did it come from? And well, one of the most accepted propositions today is what's known as the RNA World Hypothesis. The idea that before DNA and before proteins, most of life very likely relied on RNA in order to both store information and in order to create a lot of other stuff. But when it comes to this model or this hypothesis, there was actually at least one problem that was very difficult to resolve. All of the RNA molecules known to us that could actually polymerase, or that basically could copy other RNA molecules or produce proteins, were actually very large and very complex. And because they were so large and so complex, it was extremely unlikely that they would just appear out of nowhere in the primordial soup on the early Earth. So basically it was hard to explain how exactly did such a complex RNA even start. And so in this 2026 study, researchers decided to search through a massive library of different RNA sequences, actually involving 12 trillion different samples. That's trillion. That's a lot of samples. And in this massive library, they discovered a tiny molecule referred to as QT45. The RNA molecule you see right here, compared to a typical RNA that was only 45 nucleotides long and was exceptionally small for a typical molecular builder. As a matter of fact, it was at least 3 times smaller than a typical RNA. And because it was so much smaller, here it was extremely more likely to just spontaneously form just because they did not have as many units. But importantly, just like more complex RNA molecules, here it possessed very important functions. And so in this study, by mimicking conditions involving slushy, salty ice that might have existed on the early planet, researchers confirmed that this molecule can actually act as a polymerase and basically build additional strands of RNA that can then be used as a template to create brand new copies. And so basically it had what's known as synthetic self-synthesis. Although in this case, the optimal conditions involved slightly salty alkaline water and relatively cold temperatures. And that's actually a kind of a game changer, mostly because here we now have this very tiny molecule able to perform complex functions of replication without any complex machinery and without being massive, providing very important evidence for the RNA World Hypothesis, and once again suggesting that life could have indeed started with these very tiny pieces of RNA, or basically suggesting that the life-starting molecules might have been very abundant and extremely common, and were just much smaller in size than we thought. And so in more biological terms, these functional polymerase ribosomes seem to be much more common and much simpler than biologists previously believed. And that was actually an important missing piece when it comes to the story of the origin of life. Okay, so why does this matter? Or I guess what's the actual conclusion? Well, by combining these two studies on the universal paralogs and the discovery of QT45, researchers are finally able to fill this 100 to 200 million year gap between the origin of the first habitable environments and the emergence of LUCA, something that was previously completely impossible. With these studies essentially providing almost direct evidence that life almost certainly did not just appear out of nowhere and did not start as a very complex cell, but instead began with these very simple, tiny RNA replicators that eventually evolved the ability to build proteins and manage membranes, and then slowly started to create foundational systems that allowed LUCA to thrive. And more importantly, both of these studies also provide all of the necessary conditions to turn this into testable lab experiments. Something that can now be confirmed or disproven, and something that I'm sure we're going to be discussing more in some of the future videos. But for trying to find life somewhere else out there, or for astrobiology, this is also really important because here we have several confirmations that generally requirements for life seem to be kind of simple and can potentially be easily created in a lot of other conditions. And so for moons like Europa and Enceladus, and obviously for other planets out there, this is maybe good news. But until we actually go somewhere else and try to find life there, we're not going to know if this happened anywhere else. And so until future studies or until future discoveries, check out all of the links in the description. Thank you for watching, subscribe, come back tomorrow to learn something else, support the channel on Patreon where you can find additional videos without any ads and can DM me directly, or by joining a channel membership that grants you early access. You can also support this channel by buying the Wonderful Person t-shirt in the description below. Stay wonderful, I'll see you tomorrow, and as always, bye bye.